Today we will see about the G protein coupled receptors. So what is the definition and role of G protein coupled receptors? G protein coupled receptors are present in the plasma membrane of this cell, and they have a 7 helical structure which will span over the plasma membrane. And more than 50% of non-antibiotic drugs act through these receptors. We know that basic pharmacology, whenever the receptor gets activated by the agonist or antagonist, then the action is produced. So there are several types of receptors, and one of them is G protein coupled receptors. Why G protein coupled receptor? Because this receptor, which is present on the plasma membrane, is on the inner side of the membrane. This receptor is attached to a G protein. This G protein is composed of three subunits, namely alpha, beta, and gamma. This alpha subunit, it is bound to GDP. So whenever the receptor G protein coupled receptor gets activated, this GDP is phosphorylated to GTP, and this alpha will dissociate from beta gamma subunit, and then they will execute their function through other second messenger. So as we have already seen, these G protein coupled receptors, they are coupled to a trimeric structure called G proteins, and they are regulatory proteins which are bound to GTP. And what is the function of G proteins? They act as signal transducers. Whenever the receptor is bound by agonist or antagonist, this G protein will convey information from receptor to one or more effector protein. What are the functions of G protein coupled receptor? Ultimately, once they're activated through the second messengers, finally what functions they mediate? One is sensory perceptions. They regulate nerve activity and tension of smooth muscle. They are also involved in metabolism, rate and force of cardiac contraction, and they are responsible for secretion of most glands in the body. So, what are the different subtypes of G proteins? One is GS, GI, GQ, G1213. All these four belong to G protein coupled receptors. However, they vary in the way they act because they have different second messengers and also the effect they produce. Each subtype will act through different second messenger or directly through ion channels such as calcium or potassium channels. So what are the different second messengers? They can be CAMP, protein kinase A, guanine nucleotide exchange factors, protein kinase G, phosphodiesterases, and calcium released by action of phospholipase C. So coming to GS subunit, so whenever this GS alpha subunit gets stimulated, it will activate adenyl cyclase and it will cause increase in intracellular cyclic AMP. Further, it will result in activation of metabolic proteins, enzymes, and also they will cause activation of different signaling pathways, and they will regulate processes such as phagocytosis, progression through cell cycle, cell adhesion, gene expression, and apoptosis. So these phosphodiesterases, once this camp is activated and their function is done, their action is terminated by phosphodiesterase enzyme which will hydrolyze camp. So once the cyclic AMP is increased, uh, they have they act on some targets. So what are the different targets this cyclic AMP act through? One is protein kinase A, one is EPAX or exchange factors directly activated by CAMP, and third one is CREP, CAMP response element binding protein. These are the major targets. However, in cells with specialized function, this CAMP can also act on cyclic nucleotide gated ion channels, cyclic nucleotide regulated phosphodiesterases, and several ABC transporters such as MRP4 and MRP5, for which cyclic AMP is a substrate. Coming to GI subunit, It will inhibit adenyl cyclase and it will decrease CAMP and it will cause downregulation of enzymatic activity, metabolic proteins, and signaling pathways. This G12-13 alpha subunit, when it is activated, it couples to guanine nucleotide exchange factors such as P115 Rho GEF for small GDP binding proteins Rho and RAC, and they will ultimately mediate platelet function and thrombosis. Coming to GQ subtype, GQ subtype on activation causes activation of phospholipase C beta, which will increase in which will result in increase of calcium levels, and it will modulate muscle contraction or relaxation depending on the tissue which they act. So what are PLCs or phospholipase C? These are cytosolic enzymes which will translocate to plasma membrane on receptor stimulation. Once this phospholipase C is activated, they will hydrolyze the membrane phospholipid, phosphatidylinositol, and they will generate IP3 and DAG. IP3 is inositol 1,4,5 triphosphate and DAG is diacylglycerol. So this diacylglycerol, it will go and further activate protein kinase C, and inositol triphosphate will diffuse to the endoplasmic reticulum where it will bind to IP3 receptor and causes release of calcium. So when this release of calcium happens, there is calcium induced calcium release through ryanodidine receptors, and in seconds there is huge amount of calcium released, and it will activate calmodulin sensitive enzymes such as cyclic AMP, PDE, and calcium calmodulin sensitive protein kinases such as phosphorylase kinase, myosin light chain kinase, and CAMP kinase 2. In skeletal and cardiac muscle, phospholipase C activation causes contraction. However, in smooth muscle, it causes relaxation. So why does this happen? Phosphorylation of IP3 receptor by pKa causes calcium release. However, phosphorylation of an accessory protein IRAG by pKg inhibits calcium release. Though this IP3 goes and binds to the IP3 receptor, they will further activate some enzyme. So if it's pKa, it will increase calcium release. If it is pKg, it will inhibit calcium release. So smooth muscle, they work through pKg. So they will cause release of cyclic GMP, which will relax the vessel tone. So once their action is done, regeneration of G protein happens. Most of the functional activity is carried out by alpha subunit of G protein, but sometimes beta gamma subunit can exert their effect through potassium channels, calcium channels, PA3 kinase, although exact function or how they function is poorly understood. Once the alpha subunit is hydrolyzed to GDP, so when it is activated, it is in form of GTP. Now when it's hydrolyzed to GTP, it will go and bind to beta gamma subunit and become stable in activated state to regenerate the G protein, and it will go and couple back to the receptor. So here are examples and drugs that act through GPCR or G protein coupled receptor. So here we can see beta adrenergic receptors, muscarinic and cholinergic receptors, eicosanoid receptors, thrombin receptors all act through G protein coupled receptors. The physiological ligands of beta are norepinephrine, epinephrine, dopamine, and their effectors are GS adenyl cyclase. The drugs which are acting through beta adrenergic receptors are dobutamine, propranolol. For muscarinic, we know acetylcholine, it acts through acetylcholine, and here it can act through GI or GQ, mediated through adenyl cyclase, ion channels, or PLC. Example of drugs are atropine. Eicosanoid receptors, the physiological ligands are prostaglandins, leukotrienes, thromboxanes. They can act through GS, GI, GQ. Example drugs are misoprostol, montelukast. This thrombin receptors part, their ligands are receptor peptide. Effector transducers are G1213 and they act through transducers GEF, and the drugs are in the phase of development. The entire source of this lecture is Goodman Gilman, The Pharmacological Basis of Therapeutics, 12th edition. Thank you. Bye.